Okay, hi everyone, welcome to the plant nutrition video. So to start off, photosynthesis is a process that actually converts light energy to chemical energy with the help of chloroplasts. Okay, so now we need to know the word equation for photosynthesis, which is carbon dioxide plus water in the presence of light and chlorophyll will give me oxygen and glucose. Now, chemical equation you must know as well, which is 6CO2 plus 6H2O in the presence of light and chlorophyll. Please include your conditions. Will give me 6O2 plus C6H12O6. Okay, now do take note that photosynthesis occurs with the help of photosynthetic enzymes. Okay, so this is important. Now, glucose, we have also learned that is converted to different things known as the fates of glucose. So, glucose can be, firstly, used immediately for cellular respiration, okay, to release energy for the plant. Now, it can also be converted to a whole bunch of other things. For example, starch, sucrose, amino acids, fat, and cellulose. Okay, so let's look at each one of this in further detail. Now, starch is actually a storage form of glucose in the leaves or storage organs like potato. Sucrose is actually used in transport in the phloem. So whenever I want to transport glucose, you must do in sucrose form. Amino acids is the synthesis of protoplasm, okay, for building and repair. Fat is storage, such as in nuts and oil, and also in synthesis of protoplasm. And cellulose is the synthesis of cell walls. Okay, so you need to know the various functions. And moving back, we're now going to talk about the limiting factors of photosynthesis. Okay, so let's recap. A limiting factor is a variable that directly affects the rate of photosynthesis. In this case, example, carbon dioxide, light, and photosynthetic enzymes. Okay, more specifically, temperature. Okay, because enzymes are affected by temperature, so photosynthesis is affected by temperature as well. So all of this is in the equation. So if we look at the graph, we have light and rate of photosynthesis. This is something that's familiar. Now the first half, light is a limiting factor. Because an increase in the light, you can see increases photosynthesis. There is a sloping up region, right? Okay, now but after that dotted blue line, you realize that an increase in light no longer increases photosynthesis. So light is no longer a limiting factor in the yellow zone. Okay, now this same graph also applies for carbon dioxide concentration. Now there's another graph that you should be familiar with, which is temperature against rate of photosynthesis against temperature. And this graph you see is similar to the enzyme rate of reaction graph. So why is this so? Because at the start, enzymes are inactive. Then it reaches the optimum. Alright, that's when rate of photosynthesis is the highest. And after that, enzymes get denatured. So you see that sharp drop. Now, why are these graphs in these shapes? Because light is essential for photosynthesis, and carbon dioxide is a substrate of photosynthesis. So when I increase light intensity, I can increase photosynthesis. Or when I have higher concentration of carbon dioxide, photosynthesis is faster as well. So that is the explanation. Now for temperature, enzymes will actually increase in kinetic energy when temperature increases. That's why you see there is an increasing part of the graph. But after that, enzymes get denatured and lose their active site. Okay, when the temperature is too high. That is why it will drop at the end. Okay, now let's look at a common question that always comes out in MCQ is if I have this kind of a graph, all right, you can see on the left side. Now this orange color part, light is no longer a limiting factor, right? But based on the graph, I know that temperature is the limiting factor. How can I tell? Because when temperature increases from 20 degrees Celsius to 30 degrees Celsius, you see that the rate of photosynthesis increase. Okay, so at this particular point in time, temperature is the limiting factor. Now we move on to photosynthesis experiments. Okay, so based on this simple diagram, right, you can actually modify it. Okay, might be important for your practical planning. So how can I test different factors? Now for light intensity, I can simply vary the distance of the lamp from the plant. Okay, if I want to find out temperature, I can actually just vary the temperature of the water bath. Okay, by adding hot water, ice, or thermostatically controlled water bath. If I want to change carbon dioxide concentration, I will vary the concentration of sodium hydrogen carbonate solution. Okay, so that solution actually provides dissolved carbon dioxide. Now, moving on to the leaf structure, I'm sure we are all very familiar with this diagram. Okay, so please go and remember. So now, cuticle, the function of it is that it is waxy and transparent, okay, to reduce water loss from the leaf surface and to allow light to penetrate. Okay, please pardon the screen things. Okay, the palisade mesophyll. Now you can see that they are very densely packed. Normally it's 1 or 2 layers. Okay, and it has many chloroplasts to absorb the maximum light for photosynthesis. We also have the spongy mesophyll, which has a thin film of moisture to allow gases to dissolve. Okay, you realize that it is also has a lot of intercellular air space that is for gaseous exchange. We will talk about that later. Okay, now the vascular bundle, it transports water and mineral salts. This is the xylem, and the phloem will transport sucrose and amino acids. Next, the intercellular airspace. Okay, this is where rapid exchange of gas can actually occur. So you can actually write this as a part of the spongy mesophyll as well. Now the gut cell controls stoma size to allow gas to enter and leave. Okay, so please remember all of these functions. Now, palisade mesophyll will have the highest concentration of chloroplasts. Okay, so you see I highlight in dark green, then followed by the spongy mesophyll. And do note the gut cells also have chloroplasts. Okay, but palisade mesophyll will be highest distribution. Then spongy mesophyll and gut cell, they can still photosynthesize, but not as much as the palisade mesophyll. Okay, now let's talk about the intake of carbon dioxide into the leaf. So carbon dioxide will enter from the outside of the atmosphere, right? So it will go through the stoma to enter the intercellular airspace. Then after that, it will dissolve onto the thin film of moisture. So step 1 and step 2, before it diffuses into the other mesophyll cells like spongy or palisade mesophyll for photosynthesis. So remember this diagram because it will help you to phrase your answer. Okay, so now how do I put that concept into words? So first step is diffusion from the high concentration of carbon dioxide in the atmosphere to the low concentration of carbon dioxide in the intercellular airspace. And this is via the stoma. After that, carbon dioxide will actually dissolve in the thin film of moisture on the spongy mesophyll. Finally, carbon dioxide can now diffuse to the photosynthesizing mesophyll cells, such as palisade or spongy mesophyll. Okay, and this is also high to low concentration. So do highlight the keywords such as diffusion, high concentration, low concentration, parts of the plant such as intercellular airspace, stoma. Okay, so dissolve on thin film of moisture, spongy mesophyll, and diffuse. So the transport words must be there, plant parts must also be there in your answer. Okay, so that is it for the chapter. All right. Do take note. Important things is that usually this chapter, right, they will try to link it to practicals, okay? Or they can try to link it to plant transport and enzymes. So as you are studying, do try to also draw the links between these chapters. Okay, so I hope that this has helped you, and all the best studying.